Uh, while the respiratory chain pro produces the proton gradient, it also generates highly dangerous reactive oxygen species as its byproduct. As we noted when discussing the function of complex 3, the intermediate in the complex 3 function is the formation of semiquinone radical, which is integral part of the Q cycle of complex 3. The although the semiquinone radicals are shielded within the complex 3, they can be oxidized with not zero probability by atmospheric oxygen floating around, thus producing back ubiquinone and the highly reactive the superoxide radicals. This problem of generating of superoxide radicals is especially pronounced when the electron transport chain cannot function properly. For example, if the ATP synthesis cannot occur, thus building up the proton gradient across membrane, it cannot be released, and because of that, the whole flux of electrons through the electron transport chain gets stuck, thus accumulating the semiquinone radicals that can be drained down to oxygen that's generating a shortcut and constantly producing the superoxide radicals. And this could also happen when a lot of NADH generated, for example, in citric acid cycle that cannot be utilized in a timely manner. And these conditions are known as the oxidative stress. So oxidative stress is highly disadvantageous and dangerous because the superoxide radicals are highly reactive, and especially they are dangerous in the context of mitochondrial matrix, because by reacting with the iron sulfur clusters on certain components of the mitochondrial machinery, they produce highly reactive the hydroxy radicals, which can interact with everything, including proteins, lipids, and DNA, that's producing a lot of damage. Approximately approximately half, 0.2% of all oxygens that are entering the respiratory chain end up not producing water, but producing the superoxide radicals. So to mitigate this constant oxidative stress and to keep cells from damaging mitochondria from damaging its machinery, there must be a system that mitigates this constant oxidative stress. And let's discuss this system in more detail. There is a system of enzymes within the mitochondrial matrix and a homologous system in the cytosol that includes an enzyme called superoxide dismutase that use protons and the superoxide radicals to produce hydrogen peroxide and oxygen. While oxygen is not dangerous, hydrogen peroxide is still a highly reactive molecule that can damage produce a lot of oxidative damage, and this is neutralized by yet another enzyme called glutathione peroxidase that reduces hydrogen peroxide to water, concomitantly oxidizing its cognate redox cofactor, a small molecule called glutathione. So this is a thiol molecule that during oxidation by glutathione peroxidase gets oxidized and dimerized, producing the disulfide bridge. And this way the these highly dangerous superoxide radicals can be drained down to harmless oxygen and water. So the bottleneck in this system is regeneration of glutathione, so that this cofactor for gluten peroxidase in its reduced state is always available, and this is done by the third enzyme within this ROS reactive oxygen species neutralizing system, glutathione reductase. And glutathione reductase is using NADPH, the phosphorylated form of NADP, as a cofactor, oxidizing its reduced form to oxidized form. And this way, during the oxidative stress burden, the pool of the reduced active form of glutathione is used to mitigate the oxidative stress and turn into an active form, which in turn gets constantly reactivated by using the pool of NADPH. And this brings us back to the our discussion of the importance of the pentose phosphate pathway, which we discussed when we talked about glycolysis, sort of source of an NADPH necessary for mitigating the oxidative stress. So in this, the cousin of this kind of oxygen superoxide radicals neutralizing system is also present in the cytosol, and it runs on the NADPH produced through the pentose phosphate pathway. So in mitochondrial matrix, there is no no system, so there is no elements of glycolytic system. So instead, the NADPH is generated by reducing this is oxidized form NADP plus using the NADH, which is in plenty in mitochondrial matrix due to citric acid cycle as a redox cofactor, and this is accomplished by transmembrane enzyme called nicotinamide nucleotide transhydrogenase. So this way the cells can mitigate the constant oxidative stress within the mitochondria,